let's talk a little bit about mutations. And it's probably going to be it. Yeah, okay, so mutations. We figured that in RNA, the result of faulty transcription, so DNA, either replication mistakes, I should say errors, or external agents called mutagens. Now, what two types of external mutagens, like big types, you can think of?
Radiation.
Perfect. What else? That's one big source of zombie movies, right? Radiation. What else is a big source of zombies? Infections don't count, they're not really. So what else is left? I see nobody watched one of the best movies ever made called Planet Terror. No? Oh, you definitely should know. Quentin Tarantino, Robert Rodriguez movie. Oh, phenomenal. Come on. You're probably reading it when you're fueling your car. What causes mutations?
Refilling your car?
Yeah, like you're fueling the car and you're looking at the gas thing, the pump, and there's a little, in the state of California, this,
Carcinogens,
which are what? What are they, huh? What are they? No, what are they like? What are they? They're like toys or wood or plants, or what are they? Huh? Chemicals. Chemicals. Radiation and chemicals are major source of mutations.
I was thinking like octane, like
No.
their gas.
No, much simpler. Look, I have a general rule in the class. If I just ask the question, it's usually, I have a grade of from stupid to complicated. It's usually a stupid question, like 1, maybe 2. If I will ask you a hard question, I will tell it's like 7 out of 10. It's really hard question. Okay, but yeah. So chemicals, right? Or radiation. Now, how frequent the mutations in RNA versus DNA as the result of errors? RNA synthesis is bad. Enzymes that make RNA, they make errors all the time because nobody cares. That makes sense to you? Like when RNA polymerase synthesizes RNA, they can, it's not a problem. Which actually explains another fundamental feature. RNA viruses, like influenza, are very prone to mutations and to change. We learned the same about coronavirus. We thought that it was genetically stable. Turns out it's actually interesting. I'm going to save a couple of minutes at the end of the conversation to touch upon that. Okay? DNA synthesis is insanely reliable. Like insanely reliable. So the average number of mutations is 1 mutation per billion nucleotides copied. If a bacterium with 5 million nucleotides, chances of 1 mistake in 1 single copying process are less than, not even 1%. Okay, it's just super low. That makes sense. So we're going to focus on mutagens and types of mutations. So what I think we're going to do now, we're going to talk about types of mutations first. So mutations can be point and frame shift. So let's start with the point mutations. In point mutations, 1 nucleotide is replaced by another. Does that make sense? So for instance, you can have A being replaced by U. Not A, sorry. C replaced by U. It doesn't matter. You don't have to memorize which letter into which, but cytosine is replaced to uracil. Okay? So if you do this replacement, you basically replace CG pair into AU pair and AT eventually. Okay? So there are 3 types of point mutations. Silent. So there is no change in amino acid sequence. You see what I'm saying? This is why it's cool that our genetic code is degenerate. So if you change one nucleotide, but the new codon encodes for the same amino acid, then whatever. Missense amino acid is changed. Okay? Does that make sense? And nonsense. Amino acid so instead of amino acid codon, let's put codon here, you have stop codon. Does that make sense? So in this case, nothing happens. In this case, amino acid is changed, and there's a different one in the same place. Does it necessarily lead to a phenotype, to a change? Maybe not. If you have one hydrophobic amino acid, and now you have a different hydrophobic amino acid, maybe protein structure won't change. Does that make sense? Maybe it will. In case of introduction of a stop codon, oh yeah, you're going to see consequences. Because your protein will be cut short, basically. Does that make sense to you? Second one, a frame shift mutation. So this will require some explanation. So let's say we have this AUG, then C, 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 A, G, G, G. Okay? So here I made 4 codons, right? This one is a codon, and these are codons as well. Okay? So next thing that I'm going to do, I'm going to introduce a single amino acid. Well, I'm going to introduce a single nucleotide right over here. And it's going to be A. Just for fun. So it's going to be an insertion. Now look at the codons. You still have same AUG, but now check this out. ACC, CAA, AGG. Does that make sense? So the whole, it's called reading frame. The whole reading frame is shifted. This is why it's called a frame shift. Usually frame shift mutations have dramatic effect. If you're curious, insertion, yeah. Deletion as well can come to that. Does that make sense? But here's another kind of interesting story. So insertion or deletion of 1 or 2 nucleotides will make a massive frame shift, right? Now, if you insert 3 nucleotides, you're basically just introducing another amino acid, kind of, Depends on the place of insertion, of course. But generally speaking, insertions and deletions, generally speaking, result in the frame shift. Does that make sense? Frame shift mutations, they will screw up the amino acid sequence, obviously. I'm not going to go over individual, what was the word? Individual mutagens, we can do it next time. What I wanted to say a few words about